Thanks everybody for attending this session. My name is Reg Clayton. I'm a Director of Field Development at the Solvias group of companies. I'm based at the Homburg site. I'm the Deputy Site Head, where we perform cell and gene therapy potency testing. We at Solvias are delighted to be able to support this workshop, and I'm delighted to be able to share a few slides with you and outline some thoughts and some regulatory background, and to give you an insight into some of the aspects of cell and gene therapy QC in specific relation to potency assays. My background is that I'm a virologist, PhD in virology from Glasgow. I've been working in the pharmaceutical industry for about 2 decades. A range of companies from small molecule discovery through to CRO functionality, with testing of adventitious viruses, and here through to testing of cell and gene therapies. I've got a strong regulatory background, strong scientific background as well, and I really enjoy being out with the clients, getting the right scope of the work, and speaking with audiences like yourselves around the important aspects of the CMC control for the incredibly exciting drugs that you're developing. So let's go forward. We've got a short presentation of 19 or 20 slides. The agenda, there's only a few points here. That introduction that we've had, brief perusal through the regulatory framework, potency testing methodologies. We'll spend a little time there. The challenges and benefits of cell-based potency assays we'll take a walk through, and also the potency assay selection, design, and validation, which gets a lot of people's attention. We spend a lot of effort on that with Solvias. We've got some assurance strategies to review, and we've also got a small section at the end of partnering with Solvias in our capability of cell-based bioassay, particularly in the context of potency assays, to support the CMC journey. So I probably don't need to tell this audience potency testing is essential in the drug development and chemistry manufacturing control. But what is potency testing? Specifically, it's designed for testing the ability or capacity of a particular drug product. Okay, and that's guidance from the FDA here, guidance for industry potency test for cellular and gene therapy products. To design that potency testing, it's important to understand the basis of the disease, the therapeutic area, and the target populations. That should correspond to the target product profile in some way of amelioration, mitigation, or improvement of the condition. And there's an important aspect of product characterization, which comes into potency testing as well. It should be measured according to the expected mechanism of action of the drug. So often in these cases with advanced therapy medicinal products such as cell and gene therapies, we're usually talking about cell-based assays for potency rather than the more reductionist approaches where you have, typically an enzyme and a rate of reduction of a substrate in some description. Here we're usually focused on the cellular bioassays for generating the right degree of assurance around the critical quality attributes of a particular batch of an ATMP. The potency tests that are required correspond to the correspond to the regulatory framework that we've got here. We do have requirements for investigational products around identity, quality, purity, and strength. There's also stability there. And you can reference the 21 CFR 312.23A7 in that, where the release and testing of biological products should demonstrate potency specific to the product in 21 CFR. And you can see that I'm not going to read out all these names. You can read them for yourself and follow it afterwards. The analytical methods used in potency testing are usually quite, as we see from our experience in the industry, quite well defined. The biological assays are quantitative. They evaluate a product's active ingredients within a living biological system, typically a cell culture. There are instances where in vivo animal studies have been used, but there is a very strong focus on the limitation of that animal use. Okay, the Interagency Coordination Committee, validation of alternative methods. There's a very strong movement in the industry towards removal of animal studies wherever possible. And the replacement, reduction, and refinement, of course, should be second nature to many of us that have worked in this industry for a long time. We are seeing the emergence of in vitro organs or organelle um, materials that are available now. There are several companies that are making organelles available, and really interesting to see their uptake. But typically, tissue culture or cell culture systems are used in these biological assays. In the non-biological analytical assays, the surrogate measurement of biological activities, as I mentioned briefly earlier, be an enzymatic assay, reductionist approach. But we do need to provide extensive product characterization data, including immunochemical attributes, biochemical, molecular, and genetic attributes as well. So where the potency testing tends to live on the left-hand side here, or the biological assays, much of the characterization work will also fit in the non-biological analytical assays, where there's often a strong focus on molecular biology. Okay. Where we have multiple assays used, this is often in response to a product that has either complex or not fully characterized or not fully understood mode of action. And those products do exist, and they seem to elicit an array of outputs, some of which may be well understood, some of which may be worth monitoring. If a product has multiple active ingredients, of course, then you need multiple assays usually to provide a degree of control over those. There may be limited product stability. The biological assay may not be quantitative or sufficiently robust or lack precision. And in that case, you may need a number of assays to provide the appropriate degree of control around the quality of the final product or the release material. Developing multiple complementary assays that measure different product attributes associated with quality, consistency, and stability is often something we see. And typically when a client would come to us with a particular product, there may be an analytics package of anywhere between 3 and 7 assays that are designed to support the CMC journey of that particular drug or particular ATMP through the process. So there really is quite a wide range here of assays, but focusing on that potency assays, they generally tend to stick in the left-hand column, which is under the usual cell-based assays that we have. The challenges that we see in cell-based bioassays are quite diverse. In the variability that we have around materials, we have critical reagents such as fetal bovine serum and human serum albumin, and the quality of those reagents. We have quality of cytokines, growth factors, etc., that may be used in these processes and in these some of the more elaborate assays. I've seen assays go out to 28 days in cell culture for some product and even longer in others. Therefore, cytokines, etc., and stability and storage, etc., is all very important to have in terms of quality and characterization about them. In the equipment where we have multi-well plates, plate readers, and flow cytometers, there are several modalities available to us. Often we see instances where we need to transfer from one flow cytometry platform to another, which is typically not a difficulty, but it does mean that you find a plethora of analytical platforms being made available across the CMC testing platforms in different companies such as ours that have a track record of doing this type of work. We have inherent variability of starting materials, particularly biological materials and limited material for testing. That's often a challenge as well. And the stability and availability of appropriate reference controls can be a limitation. So when you're starting your CMC journey, you really do need to make sure that you have appropriate reference materials, not only in volume available at the time, but also taking into account the stability. So you can provide many batches of this material throughout the life cycle, through clinical development and through the commercial life cycle of the drug. The expertise is a huge point here in the industry. The advanced scientific expertise and time-consuming method development is not for everyone. It's difficult to build, it takes time, it takes effort, and it takes great talent. And of course, it gives you managerial issues such as talent retention and accumulation of the appropriate workforce, etc. There is often extensive training required, including when assays are being transferred from one site to another. There's a focus on consistent performance that we absolutely need to make sure the assays are running properly, but also the assays are performing consistently as well. There's a labor-intensive execution in many of the assays that we see, and the technical resource management to make sure all this runs at scale and at volume is also a consideration. I've seen many assays that are rather cumbersome, that often do not lend themselves to high throughput, or should we say high volume work into the commercial stage. So that is an important consideration on expertise and resources. The complexity of the drug system is important as well. We have increased complexity of biotherapeutic modalities, multiple active ingredients. The mode of action, as we said earlier, may not be fully understood or characterized and may have multiple pathways. Gene therapies, where the effectiveness of a gene therapy or a protein expression, a downstream effect of the transgene, may be the real therapeutic effect. But that also means that there are several opportunities in here to measure and characterize during the duration of a particular assay, and finding the right point at which to gain your measurement, which you will use as your CMC control measurement for the critical quality attribute, is often a protracted process and requires a lot of conversation, a lot of data analysis as well. And we're quite used to that at Solvias with our client base, and we actually enjoy those conversations as well. We usually get a very good output from these. So there's a lot of challenges there. Some really, really interesting challenges as well. Potency assay design and validation. Okay, there's some considerations, some criteria and validation here that I want to spend a little time with. In the considerations for this design of an assay, there should be a clear rationale for the critical quality attribute under study, and that should be very well documented. That forms an important part of the CMC dossier. A sufficient number of replicates for statistical analysis should be done during that validation, and we should have an identity and limit of sources of variability that may influence the assay performance, and that can be even as banal as the composition of the plastic of a particular inert support, such as a culture. And polycarbonate is not the same as polystyrene, it's not the same as some of the other plastics. Cells may grow differently on different plastic substrates. I've seen it happen. We have appropriate assay-specific reference materials and controls, but we also have, appropriate materials for the duration of the assay. And part of the consideration there is to make sure that they would be generated in appropriate quantity to see a drug through to the commercial stage and through to production post-BLA. In terms of the criteria, the classic SSR, sensitivity, specificity, robustness, reproducibility must be strongly featured and addressed in this validation design. And also the linearity and range is important to demonstrate as well. There's the limit of detection to consider and the limit of quantitation, and they are absolutely not the same thing. In terms of validation, we should have a sufficient description of that method with the justification and the scientific rationale regarding the validation strategy, but that should be very strongly related also to the mode of action and the critical quality attribute and how that attribute was selected. We need to apply sound, appropriate statistical design and analysis, and a considerable amount of data may be necessary. Typically, in years gone past, it was considered enough to do 9 assays typically in a validation. There may be a far greater requirement of some of data now, way beyond 9, where you do it with 2 different analysts at least. There may be some very different considerations depending on the type of assay and the amount of data that will be required here. So that does need to be very carefully considered. And the validation report, according to the GMP quality systems that we must subscribe to, the validation report, the validation summary report should have validation parameters, and those should be strictly related to the critical quality attributes that we're looking at here. The maintenance and retention of all laboratory records, I shouldn't have to tell this audience about that. We store all of that data, even from the very early stage of assay development, which is usually done in the pre-GMP space. And it starts to become GMP relevant where we have a validation protocol that's written. And a development report is also a very useful document which positions the development data as it comes towards designing the final stage of the assay upon which the validation criteria will be subject to. So all of that data and maintenance of all that laboratory records should be available even at the early discovery stage as well, which often typically outside the GMP space, but certainly supporting the justification of the strategy for assay development and validation as it goes through the GMP space. Through to potency assurance strategy. This is a science and a risk-based strategy. And the quality target product profile here should be the summary of potency-related characteristics according to the mode of action, the clinical route of administration, and the control strategy should be related to the manufacturing process and product quality. There are some manufacturing processes which do indicate a particular type of potency strategy, for instance, where and how and at what size of batch a particular product would be made, whether or not it's derived from a cell or whether it's secreted from a cell, for instance, would put you in a different validation strategy and have a different impact on a master testing strategy that would form part of that control portfolio of assays. The critical quality attributes should be attributes of the product for achieving efficacy. Where you have compliance with your critical quality attribute, you're in specification, you would expect in the appropriate disease state of a patient that you would have an effect delivered to the patient. And the critical process parameters are parameters that influence critical quality attributes. The CPPs are not the CQAs. Those process parameters can be quite diverse, and we need to take those into account when we're designing that strategy for potency assurance. The control strategy is around the CMC, control of manufacturing processes and materials, specification, chemistry, manufacture, and control. Classic control strategy. It is relevant to in-process testing, and we see in-process testing across all the biologicals, all the vaccines, and across the ATMPs. In-process testing at unprocessed bulk harvest, or in-process testing through bulk harvest, in-process testing through small batches, and through into large batches as well. It's all very relevant and needs to be considered. For lot release testing of licensed products at the drug product stage, that DP must be tested in the potency release assays as well. Okay. There is also an expectation around continued process verification and track and trend, where you have consistent batches being produced on a manufacturing cadence. Then applying these assays to it does build a database which you can actually monitor the evolution of your product, the evolution of your manufacturing processes. But the continued process verification is important, but also the assay itself and the track and trend of the performance of the assay is also very important as well. So that's a formal GMP requirement. In terms of assays, we have suitability of the assay and also the tractability of the assay, whether we can take that up to a larger scale. The control of the reference material. Often this is a well-characterized lot of drug product. Often it can be another material obtained commercially, but that control material must have appropriate degree of CMC control around it, and it must also be tested in the potency assays and be within specification and well-characterized. We do see, in fact, a lot of early stage projects. There's a concern over the level of reference materials that we have in terms of its quantity as well, and whether or not we can get batches of material. So that's certainly a consideration at these stages, what degree of volume of material, how well it would be characterized, and of course its stability and whether it needs to be manufactured on a certain cadence to keep up with demand is important as well. The assay should be qualified and validated. That can be done in a phase-specific manner, where qualification, which would be about sensitivity and specificity, would be done usually up to phase 2. And at the back end of phase 2 clinical development, the formal GMP validation would be performed. That is acceptable, but of course that should be part of your master testing strategy, and it should be discussed as part of the scientific advice at the time of the IND as well, so that you have a clear master testing strategy that would enable you to take that assay through the validation stages in a phase-specific manner to avoid any undue expense and also to enable timelines as well, where there may be pressure on timelines. The qualification and validation of assays should be finished by the end of phase 2 to support formal phase 3 transition of a particular drug. The changes and transfers that may be done to an assay during its life cycle, and it's important to remember as you use an assay more often in new batches, as the manufacturing technology may change, adapt, or the volumes may be changed, the manufacturing process may be validated even as late as phase 2, which means there may be some changes required in the CMC assays as well. So those things are important to bear in mind. They may have changes to the assays, and as phase 3 would approach for clinical development of a particular ATMP, it's also worth thinking about the technology transfer to enable the support of that drug in different territories, for instance. If you go through the US footprint and the EU footprint, it's often preferred to have testing for the release of those batches on a particular territory done in that particular territory. And of course, it also means that you have redundancy around the service provider in case of any natural emergencies, such as weather conditions or any other impacts at the local level, to make sure that the supply of drug, even in the clinical stage and through the commercial stage, remains uninterrupted. So that's a very important part. We should mention here, and I've got it as an asterisk, it should be that most cell and gene products, at least one bioassay should be included. Okay, at least one cell-based bioassay should be included. It's not enough to have a series of reductionist assays. There should be a strong focus here on at least one bioassay included. That's a very important point. Now let's take a look at what potency testing in practice looks like. At the time when, and we'll read this like a sentence from left to right, with discovery, preclinical, phase 1, 2, 3, through into commercial, the progressive implementation of a cell-based potency assay often has a number of considerations. Around discovery, we have a drug candidate selection. At the preclinical level, you have the qualification of those assays that may go into phase 1 and be ready for phase 2. We have a refined strategy based on quality and clinical data. We can see a tech transfer to a GMP site at phase 1 and phase 2. And then validation protocol, validation runs, validation report performed by the end of phase 2 to support the work going into phase 3. And then it becomes reduced to routine release testing. Okay. At the point where the BLA submission at the end of phase 3 is happening, all of those assays should be fully squared away, validated, and ready to support the appropriate volume of the drug into commercial stage. We often see it presented to us as a company that does, Solvias actually supports 6 ATMPs at the commercial stage from our Hamburg site. And we see projects coming to us at very different stages here, even as early as discovery, and some in as late a point as the back end of phase 2, where we're talking then about a technology transfer of a matured assay that may be validated or may be qualified. So all of these considerations for manufacturers and to where you're placing this CMC activity is very important, because the lead times, the transfer times, and also the training times can be very impactful at these stages. Now, I want to mention something about this company I work for called Solvias. We've got a very strong history in Europe, in and around Europe. We've got a head office in Basel, which technically not in Europe, but it's within Schengen now, so we might call it Europe. I hope I don't offend my Swiss colleagues. We have a head office at Kaiseraugst, which is just outside Basel, a little way down the Rhine in Basel. And we have another site in Basel where we do small molecule manufacture. And there's a site in Hamburg, which is around 30 minutes drive north of Basel, straight up the Alsace towards Strasbourg, where's our center of excellence in the EU for testing of ATMP products for release. Okay. We have a site in Utrecht, which is our site of excellence for next generation sequencing and molecular biology, performing genetic characterization on cell lines, and supporting the entire ATMP effort here around the quality, and also supporting us in the cell lines that we manufacture for our clients for the specific assays that we need here to support the release of ATMPs. We have a small molecule site in Canton, Massachusetts, which has a very strong focus on extractables and leachables. And we have on the bottom left, we have a research triangle park in North Carolina. We have a site there which is opening in 2025. It's actually opening next week, and I'm recording this on, I think, the 16th of January. It's opening on the 22nd of January. So by the time you see this presentation, it should already be open. Where are we now? That is the new site at our Research Triangle Park situation in North Carolina, and we're opening in 2 phases. We've got a strong focus on biologics and cell and gene therapies, and that's going to be our new center of excellence, the US footprint. We've got 2 phases that we're opening here. It's going to be phase 1, which will be open by the time you see this, and it's 20,000 square foot with 6 cell-based bioassay labs and a suite of biophysical analytics labs. And phase 2 is going to be a further 30,000 square foot addition of molecular biology, virology, and microbiology suites. And that's also going to support biosafety and genetic characterization as well. So this is a huge investment from Solvias, a massive effort for the team over the last 12 to 18 months. And that's going to enable us to support our clients for ATMP and biologics release, not only in Europe, but also mirroring that footprint that we have in the US to enable us to deal with the release requirements for both of those territories. Now, we cover the US and the EU with very complementary skills, very complementary technologies and platforms, which we think is a very exciting time for us. And I'm looking forward to being part of the team that's opening this. I've been working on it for the last 12 months, and it's a fantastic project. We're very excited in Solvias to have this opening, and we look forward to welcoming many of the members of this audience to come and see us down in Research Triangle Park in North Carolina. I want to thank everybody in the audience. That's a quick whistle-stop tour through cell-based bioassays and some of the very important considerations that we have there. I've highlighted some of the attributes of cell-based bioassays, certainly the complexity of the platforms, the complexity of some of the assays itself, and also the mode of actions and how these cell-based assays are often tuned to the different outputs that we see, such as cell death or cytokine signaling, etc. We've also discussed validation. We've also discussed our Solvias site in RTP, which is opening as a center of excellence in cell-based bioassays. And I've included here details for more information. You log on to solvias.com. You can send email to info@solvias.com, or you could also email myself, reginald.clayton@solvias.com. I want to wrap that up. I do want to say thank you to the organizers. Solvias have been delighted to sponsor this, and I thank you all very much for that, and certainly the help in preparing for this presentation, and all my colleagues at Solvias that have helped me in this so far. Solvias is on a great journey. We look forward to connecting more with our clients, and I will certainly be in the US late January and early February, so I look forward to reaching out to a few people and starting up conversations about their cell-based bioassays and potency assays to support all of these very exciting ATMPs that we're seeing reported every day in the news. So thank you, everybody. Thank you for your attention, and I'm going to sign off now. Thank you. Stop.